Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. As we return to a regular programming after a fantastic 4 series episodes honoring Dr. Marcaron, we would like to take a moment and thank our Dr. GPCR Ecosystem partners for their support. We'd like to thank Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. We have a few announcements to make. We want to let you know that we are revamping the Dr. GPCR Summit, which we ran from 2020 to 2022, and are launching the Dr. GPCR Symposia events. With this series, we aim to bring you, our ecosystem site members, regular and topic-specific virtual free live events. The first symposium of the series will be held on Friday, March 24th, starting at 8 a.m. EST. We will host a full day of talks on Zoom and a 2-hour poster/networking session on Wonder. The topic of this event is recent advances in understanding challenging GPCRs. Our confirmed speakers include Graham Milligan, Ines Liebscher, Ben Myers, Antonella DiPizio, Lucas Gratz, Jake Mahoney, Brendan Wilkins, and Anthony Picard. All trainees are welcome to come and present a poster. There won't be any poster selection. Everyone is welcome. For more information and an updated schedule, you can visit the ecosystem. The easiest way to get to it is to use the links in the footer and look for Dr. GPCR Symposia. You will be able to join us by marking your calendar and becoming a Dr. GPCR Ecosystem free site member. After the event, and if speakers give us permission to record, you will also be able to watch these videos with your premium membership indefinitely. To navigate the ecosystem and find more information about our new events and everything that we do at Dr. GPCR, please use the direct links in the footer to navigate the website. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. And today I'm very excited to have with us Annabelle Milner. Annabelle, thank you for coming and joining us this morning.
Thank you so much for having me.
I'm super excited to have you on. So why don't we start at the beginning? Can you please give us a short introduction as to who you are and what do you do?
Yes, of course. So hello, I'm Annabelle, and it's great to be part of this podcast. I'm a third-year PhD student at Imperial College London, and I'm working with Professor Eileen Haniuloglu, Professor Gary Frost, and Dr. Alistair Brown from SOC Heptares. And we're looking into a recently de-orphaned G protein-coupled receptor called HCAR1.
Amazing. And I have to take a moment and applaud you and put it out there to all other trainees, whether you're a PhD student or a postdoc, because typically I send out these invitations and people respond and say, well, I don't feel appropriate coming on the podcast with all the experts that you had on. And Annabelle, I have to say, you did not hesitate. And I said, sure, I send the invitation and you didn't hesitate. So everyone listening to this episode, when you want to be on the podcast, don't even wait for me to send you an invitation. Just email us, hello@drgpcr.com, and you're welcome to join us. Everybody has a different perspective. And I think the podcast is for everyone in the GPCR field independently from their level. And I think we can all learn from each other. And I'm going to stop
I think that's certainly true. Like there was a tiny bit of hesitation, but again, I would have liked to have seen people from the lower end of the PhD, like from the field especially. So it's nice to hear from people with different perspectives, like you said.
I think it's very important because although, yes, when you go to conferences, for example, and it's the same thing with the podcast, to pique people's interests, you need to have the veterans of the field, because yes, you read their papers, you want to listen on to what they have to say, to their experiences, but at the same time, their experiences are 40 years old and you're just at the beginning and at the time where I want to say, people like, even like me, when I started in the lab, or people like Michelle or Bob Lefkowitz, the types of we didn't run plate-based assays. That's
Exactly.
that wasn't the case. So you can't really relate or at that much, or you can't really learn something that's more applicable, I want to say. You can learn from their wisdom, you can get to know them, but it's not the same thing as hearing from your peers.
Exactly. And one thing that I like most about our lab, when I first joined, we had somebody at every level. So like a master's student, first year PhD student, second year, third year postdoc, and then our supervisor. So it then meant if you had a question, you didn't have to always go straight to the supervisor, which could, when you first start, can feel scary. You could ask somebody who's only just joined or like one person above you, especially if you have questions about assessments and stuff. And it was just really nice to have that network at each level.
Exactly. Exactly. So what's your story as a scientist? You're a third year PhD. Did you always know that you wanted to become a scientist?
So I guess going back a long way, when I was maybe 5 or 6, I had a science, one of those kind of science fun kits that you get as a Christmas present. And my dad and I would do them in the garden, obviously safety specs on, white lab coat. And he'd always make me write up my hypothesis and my findings. And I found the book the other day, and it was just really like wholesome to then see that that's what I've gone into. So I think from a young age, I've always had that inquisitive mind. Decided to do biomedical science at the University of Bath as my undergrad. And one thing that they offered that I loved, like from the course, was that they did a placement year. And so in this placement year, you could either do a year in academia or a year in industry or a year in like kind of like NHS placements. It was entirely up to you where you wanted to go. So I decided to do a year out in academia out at the Charles Perkins Centre in Sydney. And there I had a research placement looking into metabolic, it was to do like dietary influences on metabolic health. And I had a supervisor out there called Jabran Wally, and he was amazing. He had all the time for me. And it was so nice to go from kind of like, we had what felt like quite crowded lab experiences at university to then having almost this one-on-one tutoring from somebody who taught me so much more than just like the lab techniques. It was how to plan projects, and it was how to network, and then how to present your data. It was everything like that all together, and then being surrounded by other students out there. I was 19, 20 when I was out there, but being surrounded by other students that were then doing their PhD, I was like, oh, I could do this. Like, if this is what PhD life is like, actually, I am capable of doing this. So I came back and finished my undergrad, and then joined Professor Frost's lab as a research technician, got a feel for the lab, loved it, and then decided to do a PhD from there.
You made so many great points that we need to stop at some of these. One is the fact that, well, you really liked science and that you followed it. The other one is that you had this opportunity to get outside of your comfort zone, travel to the other end of the world, and also learn skills that a lot, I feel like, and I'm thinking here about myself, putting myself back into my PhD and even my master's shoes, about project planning, networking, and all of these skills you do get as a trainee, either a PhD, master's, or a postdoc. Most of the time when I talk to people, they say, well, I only know how to do this, and this is specific to their GPCR, but actually that's not true. You know how to set up a hypothesis, you know how to run a project, you know how to network with people, and that's very important for your future, independently as to where you think you'll end up or where you want to end up, whether it's staying in academia or going to industry. So kudos to you at 1920, doing all of this and then coming
in
back.
part, that was down to my supervisor out there. He was so good, and he sat me down when I first flew out there. He took me out for dinner. Obviously, I knew nobody there, and he was like, "I really want you to have a good year. I want to be more than just kind of your supervisor." And I still talk to him on a regular basis now, and we share the excitements of research and obviously the lows as well. But yeah, he was like, I want to provide you with an honest view of what science is. There's no point me giving you all the experiments that work, because that's just not what it's like. And also, like, how do you analyze data? How do you then present the findings that you have? How do you make it engaging? All those kind of like skills I don't think you, like you said, you necessarily realize you've picked up along the way. He was very good at giving me that opportunity to have that experience, and even like going to a conference. So I presented at a conference at a young age as well. So
Wow.
yeah, quite incredible.
That's amazing. And
very fortunate with that placement.
so it was a placement, it wasn't picked, but now you've also had this great experience with this mentor, which also gave you the additional insight as to what kind of mentor you wanted to work with.
Exactly. Yeah. And so I think because I then found that somebody could take the time, like he was only part of a small lab, which I think definitely helped and perhaps influenced my decision of where I wanted to do a PhD, because I found he had a smaller lab, so he had more time for me rather than getting like passed down to postdocs. And I liked having that kind of one-to-one interaction almost. So yeah.
That's great. And it also one last point before we move on to the next question is that you knew that you preferred one-to-one interactions, and that's one way of going about it, but at least you knew what you wanted, what you needed in order to make the best of that year and advance your scientific career. And I think that's also very important for trainees before you get into a new lab or a new job, to figure out how do you work best and know yourself.
Yeah. I think at that age, being so young and so new to science, we'd had the very basic lab practicals at uni, where you'd learn how to hold a pipette and add protein into a Western blot, but never like I said, like experimental plan. Why am I doing this? How am I doing this? How to optimize? And so by having that kind of one-on-one supervision quite young, it was like all these questions that we now ask as regularly as a PhD student. But instead of just being like, oh, it's not working, what can I do? He kind of trained me to think, okay, take a break, let's research, let's think about the next step. And we could work things out together. Whereas I feel if I was perhaps part of a bigger lab, I would have been given experiments. And if they didn't work, they didn't work. And we wouldn't work things through. It'd be down to the supervisor to work through, rather than having that like wholesome experience of like I said, the highs and lows of research.
No, agreed. And yeah, taking a break when something doesn't work is very important. You can just
Definitely
drop everything and go home, do something else and come back with a fresh mind. Because hammering at the problem while you're still frustrated about the thing not working is not necessarily.
Help.
It's
and
not
you get
healthy.
more frustrated. For sure.
Exactly. So how did you get into studying GPCRs?
So the placement that I had out in Australia was looking at metabolic health in relation to diet. And I found that really interesting, trying to see, we worked with a mouse model, and so seeing the overall effects. So we were looking very much at downstream markers of inflammation, of obesity, of diabetes, things like that. And I've always been quite interested with mechanisms, I guess, like why does that happen? What causes that to happen? If I change this, what's the knock-on effect further downstream? So then when I did my research technician job, although it wasn't for particularly long because COVID had happened, that was then moving into gut hormone release. And as we know, gut hormones are secreted through some GPCRs. And so that then kind of put my interest then into dietary influence on gut hormone release. And then my work at Island's lab, which focuses then on GPCRs, is then looking that little bit further upstream at then how these GPCRs result in influence in the gut. So yeah, I think it's very much the drive for understanding the mechanism from kind of, I guess, like a ligand activating a receptor, then what happens? If a different ligand activates the receptor, what's the downstream effect from there?
That's great. And did you, when you first learned about GPCRs, what was your initial thought? Did you feel like, okay, I can ask all sorts of questions around this family of proteins, and the main question for you is the mechanistic, is why does it happen, or how does it happen? Was there any aha moment where when you first heard about GPCRs in this context? And no is also a valid answer.
No, I think there was. I think I'd always approach GPCRs from the mechanistic side of view. So if a ligand binds, what's the downstream effect? But then being in a lab that focuses solely on GPCRs, we all work on very different aspects of GPRs. So we have people that do the signaling aspect. We have people that do the structure and dimerization, oligomerization. We have people that then look at like spatial temporal control. So it's all we all look at GPCRs, but in a very different way. And I really like that. And then obviously we all like share the knowledge, and we can all then look at our own GPCR through these different like aspects within the field itself. So it's actually quite a broad field, which I think when I learned about GPCRs at uni, I'd never really appreciated it because it'd been delivered to me in such a way that it was the signaling mechanisms, proteins are recruited, causes downstream effects, not all the other interactions and the variability within what can actually happen.
Yeah, I feel like it also depends on who's teaching the class. So that's what happens. All right, great. You mentioned metabolic diseases and hormone controlled events. Any favorite GPCRs in the bunch?
I would have to be very biased on this, and I've only looked into HCAR1, which is the receptor that I'm studying for my PhD. So yes, that would be my favorite GPCR.
Can you tell us a little bit more about it?
Yes, so it's quite a relatively new G protein coupled receptor. It was de-orphaned in 2008, where a team out in, I think, California had shown that this receptor was activated by lactate and had a role in inhibiting lipolysis. And kind of the state of the literature before that had showed that lactate was very much just a waste metabolite from glycolysis, whereas by identifying a receptor that the molecule could bind to, then kind of gave it a little bit more purpose, I guess. So there are other nutrient-derived metabolites, such as like the short-chain fatty acids that can bind to other GPCRs, and that's very well known, whereas lactate-activated HCAR1 is less known, and I think that just adds to my excitement, at least, studying it.
That's so interesting. And when every time you feel like, oh, we've identified most of these receptors and we know what their ligands are for the majority, then a new one pops up, and then you realize, oh, this thing we thought was just discarded actually activates a GPCR.
Exactly, and especially with lactate, which was only considered as a waste metabolite for so long. And actually, It has a whole range of signaling processes involved in lots of different kind of diseases and modeling states and things like that. So yeah, very interesting.
It is very interesting. So you mentioned it was de-orphaned in 2008. What does this receptor signal to?
Sorry, could you repeat the question?
Yes, of course. I think there was a connection issue. I was just wondering, what is the signaling mechanisms by which lactate activates HCAR, and is it coupled to G proteins? Does it recruit beta arrestin?
So yes, the work so far shows that lactate, predominantly L-lactate, although we are finding that D-lactate is binding to HCAR1 at quite high concentrations. But lactate activates HCAR1, which then signals predominantly through a GI-coupled protein, and then inhibits cyclic AMP levels from there. There's also some work done to do with like the MAP kinase pathways, and it is shown to activate ERK and then MAP kinase and things from there. And also beta arrestin, there is some work, I think, in macrophages to do with HCAR1, recruiting beta arrestin signaling, and that's something we would like to look at. If we knock out beta arrestin, what effect does this have downstream?
Wow, I think it's fascinating to have, lactate. Who would have thought? And then there's a difference in D-lactate versus L-lactate. I mean, it's just really fascinating. You mentioned metabolic diseases. What kind of diseases is this GPCR involved in?
So it's involved heavily in cancers. Obviously, the tumor microenvironments have high levels of lactate, and the lactate is activated through the receptor, and the receptor then signals and promotes angiogenesis and then cancer cell survival. It's also involved in neuroprotection to some extent. So I think there's been a really recent paper out which showed because it decreases intracellular cyclic AMP levels, this reduces the phosphorylation of a protein called tau, which is involved in the neurotangles. Yeah. So I think there's potentially some work going in towards HCAR, perhaps like stimulating HCAR1 for Alzheimer's treatments. My real interest, I guess, in like a disease state for the receptor that I'm looking at, by looking into the gut, you obviously get a lot of inflammation. And based on what we eat and the gut microbiota, you get a lot of fluctuations within lactate levels itself. So by then looking at, obviously you can get some good inflammation. So you've got like a small intestinal injury, you would like some inflammation to keep the gut in homeostasis. However, too much inflammation then causes things like inflammatory bowel disease. So it's kind of that interplay between, I guess, the two. And by understanding then, well, my interest is in understanding the differences between perhaps what L&D lactate can cause for the inflammatory markers through HCAR1.
Wow, I think I still cannot believe that lactate actually binds the GPCR and then activates it. But to be honest, this is the first time I'm hearing about HCAR, and I think it's just phenomenal. I think after our podcast recording, I'm just going to jump on PubMed real quickly to get a better sense about the receptor. And so you mentioned it's a more novelly identified receptor. In your daily lab work, what type of assays do you typically run? You mentioned animal models, but what does that look like from your perspective, the types of assays that you run?
So at the moment, for this project, I'm mainly working on cell models. So I've got CACO2 cells, which are cells that model adenocarcinomas from the colon. And then we'll so I'll treat them with L&D lactate and kind of see the differences from there, and then look at kind of downstream markers, whether it's things like cyclic AMP levels. We did look at IP1 levels to then rule out GQ signaling. We've done a lot of flow cytometry to kind of look at whereabouts the receptor is expressed within the cell. We've done spatial temporal control, so looking at co-localization to markers in different membrane proteins, such as like early endosomes and very early endosomes. And now I'm beginning to look at the inflammatory markers as a downstream result through Western blots.
Yeah, so you get to use a variety of tools to study the receptor in these cell lines. Anything that you wish you had, if you had a magic wand and you wanted to learn more about your favorite GPCR, what would that look like? Without wanting to put you on the spot here.
I think something that would be really cool, if was possible, would be to kind of create like an organoid system of the gut, because obviously that exists, but the microenvironment that the receptor is expressed changes so much depending on dietary intake and then time of day and then genetics. So if you had an organoid to express, like that can be exposed to the different fluctuations within the microbial products that are produced by the microbiota, and then look at things like cell impedance, which we don't have here, to then measure like kind of, I guess, the crosstalk between different GPs, the overall so cell impedance measures the overall outcome of the cell. You'll pick a marker and it will measure that. So then there's a lot of different GPCRs that are expressed within the gut, and they must be crosstalking and all reacting and being activated and deactivated by all the different ligands at the same time based on what you're eating. So I think kind of, because it's so dynamic, it'd be interesting to have one final output reading to then understand, almost going backwards, what's happening, where's the crosstalk happening between these GPCRs. So I love it. That'd be quite a cool model.
I love it. And who knows, maybe someone listening to this podcast episode will reach out to you and say, well, I can help with that. We do this on a regular basis, or two people. You never know. I think I like to ask this question because this allows, oftentimes, at least, again, I am the starting point based on which I blurb these things out, but I try to tell myself, okay, if I had no obstacles, what would that project look like in 5 years? And it's a difficult thing, because as scientists, we're always trying to identify what are the roadblocks and what can we versus what can't we do. But I think it's important to tell yourself, okay, if nothing is a problem, what does that final experiment look like? Or what does that project look like?
And it then allows for a lot of collaborations, because somebody might, for example, know a lot about gut organoids, and I know that they are a thing and a lot of people use them, but then pairing that then with a cell impedance technology, kind of putting it together would then allow for the sharing of work.
Yeah, so who knows, maybe someone will reach out, we'll have your information on the site, and who knows. I think the GPCR field in general is very collaborative, and people are always happy to help, unless they're super busy. But I think it's something that could happen. And it would be, and if it does, please let me know. That's why I'm pushing this right now, because if it does, please let me know. This is our goal at Dr. GPCR, to really increase these collaborations. Nothing makes me happier than to hear, oh, I talked to this person because I heard about them on your podcast, and we ended up collaborating, and we have a paper together, or we're starting a company together, or you never know where that can lead.
Yeah, exactly. And this is all from your platform that you've provided. So thank you.
Of course, that's also in some ways an answer to the needs that I had as a student, where I spent months researching the vial of HIC cells that was used in a previous publication that I needed to use, or going around and asking these questions, but not necessarily having the tools. Well, we had email. That's good. I'm not that ancient, but I think about, people 2-3 generations before me, where you had to write letters and send them through the mail, and picking up the
this
phone.
allows very quick kind of collaborations, like you were saying, and it also then makes people feel more approachable. From I guess I'm quite new to this field, but by hearing these podcasts, it's really nice to then be like, oh yeah, that's that person, I've listened to that podcast, I know more about their work. It's quite an easy format to listen to people's work as well. So you can put it when you're on the train or something, instead of sitting there trying to read papers all the time.
Exactly. And then tying it back to what you were mentioning previously about learning how to network. You meet one of our podcast guests at a conference, and you can go there, introduce yourself, and say, I love your podcast. And there you go. And now you have something to say. Because you also mentioned, whenever you're more junior, approaching more senior scientists is a little bit intimidating. But starting off with, hi, my name is XYZ, and I loved your podcast on the Dr. GPCR podcast, is a nice way to break that ice, and then have conversations around a poster, a presentation, you name it.
Yeah, it certainly makes people feel a lot more approachable. So
that's the goal. And I also find a lot of times, although people tell me you can't hear it, but I oftentimes, whenever I was interviewing more senior GPCR experts on the podcast, I felt nervous, but now they're completely approachable when you hear their success, but also failure stories, which is, I think, very important to point out, that we all have that failed experiment, or that series of failed experiences, which make
definitely, especially as I guess it's a young scientist, you always get this picture perfect image of every experiment, especially when you go to a conference. Obviously people are presenting the data that's worked, and so you get this kind of tainted image of all the experiments always work all the time, and that's not the case in science. That's not at all. And so like you said, really nice to hear.
Yeah, and I think we should have these slides of the list of people who worked on experiments that did not work, which enabled then figuring out what the protocol is for the experiments that actually worked.
And did work. Yeah, you kind of need to credit all the work that didn't work as well.
Exactly. I'm gonna ask it, do you think GPCRs are still good drug targets? I think the answer is always yes. At least it has been so far. I'd be very surprised if someone would say no. But any additional thoughts around that?
I think obviously, yes, that's why I'm working in the field, and also partnered by a company that specializes in GPCRs. I guess as we get more and more knowledge about them, and especially as we're finding more and more receptors and understanding how they work, they become more and more important, because it's not always just one receptor target. Or if they dimerize, if they traffic to certain locations, the more you understand, the easier it is to target, I think. And there's so many new advances in technology, especially like protein, like cryo-EM and protein structure and things like that, that I have no knowledge of particularly, but I know that it's very useful for targeting GPCRs. And so, yeah, my answer is
I think you made a great point, and tying it back to your idea or your wish about studying HCAR in organoids and looking at how it interacts with other receptors. One thing that I think is still missing is, well, there's two major things. One is really the link between, receptor ligand and what happens in the cell from a disease perspective, really mapping out and really knowing no matter what cell you're looking at, what's pathways activated, but then and also that relationship between the different activation, the activation of different GPCRs. And although I live in the GPCR world, there's other proteins out there that also play a role in cellular function. I think having that holistic image is still far out in the future.
Yeah, I think it's nice to appreciate that there are obviously other types of receptors within a cell, but we can both be biased and love
I think so too. I think so too. We've talked a little bit about you traveling abroad and then getting this brand new experience working in a lab, having a great mentor that not only taught you how to think about a project, how to plan a project, but also got you thinking about presenting the data, which I think is very important, and also how to network. Any advice that you would have for junior scientists or your peers?
I think for me, having that placement year was critical in deciding that I wanted to continue within this field. I went to uni, and a lot of the uni course structure was a lot of lectures, and I'm very much a hands-on person. So I think through my time at uni, I debated whether this was right or not. I loved the content, but I wanted something more practical. And then by having that year out and really experiencing research for what research was, and being surrounded by other people kind of my age, and they just started their PhD, to people that were PIs, professors, seeing that kind of career path really helped me make that decision. So I guess my advice to undergrads would be, if you can, try and get a summer internship or even just a 2-week placement shadowing. Reach out to people. Somebody will, I'm sure, offer you some experience, and then it allows you just to ask all your questions that you have, like what day-to-day life is, or what do I need to do to try and get a master's if I want to do a master's, and it starts to build up those connections that you have. And then I think the second piece of advice, I guess, aiming at even younger people, would be, because so much is now online due to COVID, you've now got a lot of people that are online themselves. So like a lot of people have a PhD Instagram account or a PhD TikTok account where you can see almost like snippets of what their life is like. And that may feel a lot more approachable for you to just to follow for a bit, see if it's something that you enjoy, or like reach out to that person, because it's probably somebody your age. That might seem a lot less scary than contacting PIs, and we all know PIs are very, very busy, so you might not get a reply, which is something you have to bear in mind, but that doesn't mean to say you won't get a placement either. So yeah, I think for me, that hands-on experience of understanding what it was actually really like probably shaped my career a lot. So my advice would be to network as much as you can, contact people, ask your questions, and then if it's not for you, and you've not wasted 5-6 years going down that route and finding out it's not for you, you can decide within a couple of weeks whether it's something that you want to continue or not.
I think that's very valuable, and a lot of people in my class during undergrad, we had either a summer internship that you could do, or you had to do it during the school year, and it was 4 weeks. And a lot of people very early on decided, oh, this is not for me. Lab work is not for me.
But at least you can make that decision quite early on then, rather than leaving uni thinking that's what you want to go into and then deciding it wasn't.
And then feeling miserable for the next 5 to 10 years, trying to do something that you don't really like. So I think it all boils down to going out there, trying to have as many experiences as possible, trying out lab work, trying out other specialties, and then decide whatever works for you. How did you, so based on your first experience during that year in Australia, how did you pick your PhD lab?
I guess it was, so the work that I did out there was quite nutritional based, and it was the consequence. So there was, sorry, going back, at university, we had a whole list of like different placements we could go to. You still had to be interviewed by the supervisor, write a statement, as like apply for it like a job, and then the supervisors picked you. So I was then picked and I went out to Australia, and that lab was very heavily focusing on metabolic health and nutrition. So then coming back, finishing my degree, I wanted to get a job, and I really enjoyed that metabolic kind of pathway research. And the skills that then I'd picked up from that lab very much overlapped with the skills that Gary wanted for somebody as a research technician looking then into gut hormones and metabolic health. So there's a lot of overlay there between the two. And then Gary does a lot of work with Eileen from like the nutritional GPCR point of view, and a PhD came up and I applied, and that's kind of then how I moved slightly more into the GPCR field.
Now, I was asking the question because I think getting a previous lab experience and realizing that you like it also gives you an additional layer of experience as to figuring out how that first environment was. And you were very lucky because you had a supervisor in Australia who spent time with you, who taught you things. And I think that really helped facilitate you making the decision of, applying for a lab-based job and then switching to a PhD. So there's multiple things. Please.
That placement also allowed me to decide what parts of the so I knew I loved the lab side of things, but because it was quite a varied project from working with mice models to cells to then metabolomics, it allowed me to decide which specific parts I enjoyed most. So I didn't particularly enjoy like the bioinformatics so much. I knew if I was going to pick a PhD to kind of move away from that side, but I could still find a project I would like. And I guess with a PhD, you can kind of shape it, obviously within reason and interests between you and your supervisor, you can shape it to the directions of what you enjoy learning about and wanting to explore further. So yeah, I knew that I wanted to move away from bioinformatics, but more into the downstream signaling and out there through outputs.
Yeah, and that's also very important to know that, yes, but what I wanted to get to, and please excuse me blabbering, it's still not even 7 a.m. here, is the fact that your first experience in the lab will form you, will make you decide whether you like it or not. But in case your first experience is not optimal, and you still like the lab, you need to be able to separate the human factors of that experience and the scientific factors, and then figure out what is the type of environment that you like to be in. Some people prefer a big lab with 40 postdocs and you just, interact with postdocs. Others people prefer a smaller lab, and that all stems out of what fits you as a person.
Exactly. And it can also depend very much on what field you're in as well. So you may really enjoy the techniques, but you're struggling with the literature. But the techniques are transferable between a lot of fields. So everybody does Westerns and QPCRs, and that can at least be transferred. So even if you don't necessarily like the lab, if you like what you're doing, keep an open mind that it could just be that the lab you're in does not suit your interests. I wouldn't close the door completely based on one experience.
Exactly. Plus, it's okay to first go for a job and then decide that you want to go to grad school and do a PhD. And, as long as the road that you're again, what I'm trying to get to is that, at least from my perspective, when I was growing up, scientifically speaking, the thought was you do a bachelor's, then you do a master's, then you do a PhD, then you do a postdoc or two, and then you become a professor. And what I'm trying to get to is that actually this is not true. And in today's world, you can get to a PhD program from all sorts of directions. And it's okay if you want to take a pause, or if you want to take a detour, or even if you start a PhD and a year in, you're like, oh my God, I hate it. or I don't like this environment because this environment is not for me. And then you can give yourself a break and be like, okay, let's take a break, exactly the way when you would do it when an experiment stopped working or never worked, and then figure out what works for you.
Exactly. So I always thought that you had to do like get a first in your undergrad degree and then do a master's to be able to do a PhD. But that's not necessarily the case. You can get your experience in industry and like build up your experience and then come in. You don't have to do the logical normal pathway to become in. And one piece of advice that I was given was don't be in a rush to do a PhD, like get to know a lab before you commit to 4, 5, 6 years of your life in a lab, which is why I guess I took up that position with Gary, because it allowed me to work. I think the contract was for a year, it allowed me to work for a year within the lab, see how it functions, see if although I knew I'd like the area within the lab, did I like the lab itself, how it was run, everything like that. And I did, obviously, that's why I took a PhD. But it then supervisors are such a big part of your PhD life that you obviously want to get along well with a PhD, with your supervisor, sorry, as you are with them for quite a while. So yeah, I guess another piece of advice that I would say is if you can try and perhaps do a small position with them before committing to a PhD, so then you can actually speak to the supervisor, get to know them before just jumping straight in.
I think it's important, and also speak to the people in the lab. And also find people who were in the lab but are not in the lab anymore, to get the full picture. Because the other thing is that I think PIs over time, they change, because their perspective changes, but also their level of experience changes
and
as
demands
well.
of other things that they're committed to.
Exactly. So I think having a more holistic image as to who the person is, how does that person work with their trainees, but also how that person was 5-10 years ago, gives you this idea of how the PI evolved over time, which allows you to make a more informed decision.
Exactly. Yeah.
All right, last question. Top 3 aha moments that you had as a scientist that shaped your trajectory. Anything you feel like got you to where you are today?
Okay, so one of them would be out in Sydney. I presented work. It wasn't at a conference, it was just in front of a very large audience. Probably there's a whole building thing, so maybe 400, 500 people. 1920, I hated presentations, was so nervous doing them, absolutely hated them, would dread the day until it came up. But I think standing there and presenting work that I knew it because I'd done it, and it wasn't just a random topic given to me, and actually like seeing the audience follow and being engaged, and then the discussion afterwards. And sometimes you can answer these questions and sometimes you can't, but actually having people interested in what you've done and something you're passionate about presenting, that just felt like such a nice rewarding moment to me, because it was such the flip between hating presentations to actually, I didn't hate presentations. What I hated was not knowing what I was going to say, but then talking about something that I'd done day in, day out really helped. So yeah, doing that presentation, quite a young age, quite a big audience, was huge to me. And that's when I was like, oh, actually, if this is how we talk about science, I really enjoy this. So that's probably number one. Earlier this year, I took part in a conference called the Society of Endocrinology Conference. And I presented my work, again, another presentation, but I presented my work and I won a prize. And so that was really nice, like nice to be recognized for the work that you were doing. And again, just felt so wholesome to get that feedback, to be like, oh, you were really good, we really enjoyed listening to your work, can we read anything more about what you've done, and those kind of questions. And even more recently, on a lot smaller scale, there was an experiment that I've been doing for trying to get to work for like 4 or 5 months, and it just hadn't, and we tried all these different ways of optimizing it, and finally after 5 months of work, it's starting now to work. So tiny aha moment, but it felt really good.
Yeah, it does. Thank you for sharing that. You mentioned presenting and being stressed out before giving presentations. Any tips or tricks that you want to share as to how do you get the audience engaged, and how do you basically calm yourself and just do what needs to be done?
I think to get the audience kind of involved, you've got to be confident in what you're saying, but also deliver it as if you're having a conversation kind of with a friend. You wouldn't speak very monotone to a friend. So like come alive with the presentation helps a lot. If you're just stood in the corner, very shy, very tiny, it makes it a lot harder for people to sit through 20-minute talks back to back. And then what was the other question? Sorry. How do I prepare?
Yeah.
I do like to normally write out my script beforehand, read it through a couple of times, and then not touch it. And whatever I've remembered are then the key points that I need to take and present the work. So I will practice it once or twice, but don't ever over practice. Otherwise, then it won't sound natural and it won't flow. And if you miss a line out, you're going to lose your place and you'll fumble. So kind of take away key points from what you've remembered and deliver it from there.
That's really valuable. I do the same. I always write out what I want to say, but then practice it and then not look at the notes anymore. Because as you mentioned, I've tried it the other way, and then you forget the word or the sentence, and you know
the one key word to lead into the next step.
Exactly, and then you're stuck. And I think the other thing I would add to this is practice, practice, practice. So any opportunity you get to give a talk, to present, to talk to people, is one additional opportunity for you to get more experience.
I actually find the worst presentation to do is when you practice in front of your friends, because if you can get over the giggles from like presenting in front of your friends, you'll be fine in a room full of strangers. So that's my harshest critics. Not even in the field, but if they sit there and they listen to me present, if I can do that, I can do it to a big group.
That's amazing. Thank you, Annabelle. I had such a blast talking to you.
Well, it's been so lovely to meet you.
Absolutely. Thank you for coming. I forgot to mention before we started recording, stay here after we stop recording. So anyone listening to the podcast is going to wonder, what are we talking about after we stopped recording? But that's going to be between me and Annabelle. Thank you again. And I want to say, thank you for being here, but also thank you for not hesitating to book this podcast episode. And putting it out there for
opportunity.
Of course, and putting it out there for all PhD students and master students and postdocs. Everyone is welcome. And it's an additional opportunity for you to talk about your work. And it's in a way a presentation as well. And you've been amazing.
Thank you.
Thanks, Annabelle. Bye-bye.
Bye.
Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest, as well as take a moment to thank our team members, Attila, Ines, Monse, Ivana, Andreina, Balint, and Julia. A huge thank you to our ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem, so don't forget to join the Dr. GPCR ecosystem today. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you're like our podcast, leave us a 5-star review on Apple Podcasts. You can also leave us a testimonial by following the link to the testimonial forms in the ecosystem by using the footer. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions and suggestions at hello@drgpcr.com. And until next time, stay safe.